Welcome to those who are new to Malaysia Research Insider. And to those who have joined us last Saturday, welcome back. I'm Apple, and I'm doing a PhD in Edinburgh in plant sciences. So this science chat is co-organized by MBIOS, 100 Scientists of Malaysia, along with our media partner, Science Media Centre Malaysia. So let's welcome our scientist today, Jake, who's a first-year postdoc researcher in Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. So I'll let Jake introduce himself further. So over to you. You can share your screen.
Okay. So I'll be talking to you about a really interesting protein in my opinion, and that's ubiquitin, and how it relates to health and disease. So I'll try to make this as painless as possible. So I will talk a little bit about my science during my PhD that I've done for the first 4 years, in the past 4 years. And then I'll talk about my postdoc, which, to be honest, has only been a few months. It's not that long. And then I'll talk about my journey rather than talk about where I started off in the beginning. I'll leave that at the end because I feel like then people have more attention. So I lived in Malaysia up to 2013. Then I moved to Sydney in 2013, and I've lived in Sydney for 7 years, which sounds like a long time, and it is. And now I'm in Melbourne, which is not also in Australia. So I also want to thank the people organizing this, because I realized that organizing an online event is actually not easy, because you have to manage times and then you got to get people. And although it's cheaper, that's for sure. So I do want to thank the people for making this possible as well. And Apple for managing to find me on Twitter somehow. All right, cool. I won't talk too much about the science, but if anyone's really interested in the heavy science, you can just go to YouTube. I've given 4 talks already this year, so I'm a bit overwhelmed. So you can look it up if you want to on YouTube, and you can go to the American Society for Biochemistry and Molecular Biology to look for more YouTube videos, or you can go to my blog to look at my science content. So today I'll just kind of give you a little bit of a brief overview. Cool, I'm just looking at my time. So I'll talk to you about my PhD a little bit, and really asking about how do we control how much cholesterol we make in our body. So I'll tell you why this is important, and I'll give you a bit of an introduction. I am trained in molecular biology, and what this means is that you have all these different levels of life, and basically you have the human being, and then afterwards you can go down deeper and deeper. You have things like your heart, your blood vessels, your tissues, like the muscle tissues. And I really am a molecular biologist by training, so I work on molecules, things that you cannot see by eye. And so the molecules that I worked on mostly in my PhD, as many other people in the WhatsApp group actually, is proteins, DNA, and lipid. A lot of people might not want lipids, but I was interested in cholesterol, which is a lipid molecule in our body. So in my PhD, I worked on all these things, but the lab I was in was very interested in cholesterol. So, but it's not just that, beyond the organism, there's actually more to that, that we have the ecosystem, the landscape and everything. And there are people who study the land and sea. You might have come across Stanley's work in the past few weeks. If you haven't, go check it out. He studies magmas and volcanoes. So there's really a lot more than just, us as one person. We live in a community. Okay, cool. So cholesterol looks like this, right? How do we control cholesterol levels? How do we get cholesterol? You get cholesterol from your diet. About 20 to 30% of your cholesterol comes from your diet every day. And 70 to 80%, you make it by yourself in your liver. So that's a lot from yourself. You don't actually eat that much of it. And your cells in your liver can help make cholesterol. So there are these cells in your liver that help make cholesterol. Why is this important? Why do we need to care about this knowledge of how we make cholesterol? Well, you might know someone, chances are you might know someone really, that take these drugs called statins. And so statins are drugs that block your cells in your liver from producing cholesterol. And this is a very big market, worldwide, billion-dollar industry. So you can block your cells from making cholesterol. So this is one way to lower cholesterol levels using a drug. And cholesterol is obviously very important, but at the same time, very bad for you. Too much cholesterol is associated with heart disease and cancers. Too little cholesterol can lead to growth and developmental defects. So there are syndromes where if you don't have enough cholesterol, you're born with developmental defects. You need cholesterol to grow and be strong and healthy. I won't say too much except for the fact that low cholesterol is being linked to antiviral immunity, that this is actually kind of an interesting area of research, but I won't comment too much on that. But basically, we need to control how much cholesterol levels we have in our cells if we want to be healthy. In your cells, you make cholesterol, all right? You make cholesterol at the end here, but you need many, many steps to go to cholesterol. There's a lot of small steps. And these steps are made possible by these proteins. These proteins, and I'll tell you the name now, but they're very important because I'll revisit them very soon. But these two proteins are called HMG-CoA reductase and squalene monooxygenase, and they're in your cells. So the statin drugs actually block HMG-CoA reductase. And if you block this, then you cannot make cholesterol. So these are rate limiting steps, and I'll explain to you soon what rate limiting actually means. Imagine that you have a factory, right? You're trying to make cholesterol at the very end. You have this guy and this guy. They're called HMG-CoA reductase and squalene monooxygenase, and they're very, very slow sometimes, or they're very fast. What it means is that they can control how fast or how slow cholesterol is made. So if this guy packages things very slowly, then everyone else later on has to wait for him. Or if he makes things really fast and packages it really fast, then everyone down later on, well, they're forced to be faster as well. So these two guys, HMG-CoA reductase and squalene monooxygenase, they can control how fast or how slow your cells make cholesterol. So that's a natural process that you use to control cholesterol levels. And I'll tell you how this works soon. Cool. So when you have this enzyme or this protein in your cells, sometimes you can have enough of this molecule called lanosterol. It's somewhere in the middle here before you go to cholesterol. When you have enough of this lipid molecule, you can actually join HMG-CoA reductase to this thing called ubiquitin. And ubiquitin is like a signal. It then recruits and tells the cell to bring this gigantic protein to come and chew it. So it will come and chew it, and then this destroys HMG-CoA reductase and prevents more cholesterol synthesis. So this is a natural process that your cells can use to control cholesterol levels. So ubiquitin is like a name tag. It's like a name tag that says "eat me," but it does many other things as well. So this is called ubiquitin. But then it's not just HMG-CoA reductase actually, and my PhD supervisor and his team later on showed that you have another step that can be controlled as well. It's not just one step that controls everything. So similar, you have a lot of cholesterol in a cell. This will make squalene monooxygenase join to ubiquitin, and then this gigantic protein will come and chew it. And so when that happens, it destroys this and you lower cholesterol levels. So this is natural. And this was, I didn't show you the reference because I didn't quite show the data, but this was discovered by my PhD advisor when, I think in 2011, way before I joined the lab. Cholesterol metabolism is very complicated. I just only show you a really, really, really small snapshot. I won't go through too much details, but there's a lot more than just how we make cholesterol. There's like how you transport it and how you store it. So I won't talk about that. This is where it's a little bit complicated, but this is more for the scientists, I guess. And but I'll try to make it as simple as I can. So in my PhD, I was trying to understand how this process happens, how cholesterol can make this ubiquitin join to squalene monooxygenase, and then this destroys it and then blocks cholesterol production. So this is what we call basic science research. And what this means is that it's kind of trying to discover something. It's not like we're trying to cure cancer or cure heart disease or something. It's really, you have this question that's curious, and then you just want to go and find out how it works. Okay, that's science. Now, squalene monooxygenase has two parts. one part is called a regulatory domain, and this is where cholesterol can come and we think it binds. We think cholesterol will come here and signal here to be joined to ubiquitin, and then this protein can come and chew it. So I was very interested in understanding how this regulatory domain works, because this is very important to control how much activity is there to then control cholesterol synthesis. So this is what the regulatory domain looks like. That's from my PhD work as well. I did some of that. So this is where it's a bit really heavy, but I'll just tell you what I spent 4 years doing, basically. Normally I will be able to see the questions, but oh well. So my PhD work was, like I said, trying to figure out how it works, right? So I showed that in this small part of squalene monooxygenase, there's a helix, and when this helix is present, it can actually sense the cholesterol in the membrane. So this is what we call a lipid bilayer. It's found in your cells. Cholesterol lives in there normally. So I found that there's a helix that's in the membrane, and later on, when there's a lot of cholesterol around this bilayer, this can be deformed, and then eventually it becomes joined to ubiquitin on these sides. And later on, I showed that there's a protein called VCP. It pulls it out. It literally pulls it out of the membrane, and then it gets destroyed outside. So that was basically, you know, like my 4 years of work, and each of them was published in a different story, I would say. But yeah, that was basically it. That's my 4 years in 20 seconds. Because I won't go to too much details, but just to give you some idea of what it looks like, how I discovered this in a way. So what I did was I just had this portion, like just this part. You can see that I can take it and fuse it join it to a different protein, like green fluorescent protein, which is GFP. Basically, you just need to see if I add cholesterol into a cell, there's less dark band, which means this guy is being destroyed. But when I chop out the helix, what you can see here is I chop out the helix, I add cholesterol into the cell, this guy is no longer destroyed. The band is the same thickness. So that's telling me that I found this region, which is very important for knowing how much cholesterol is in a cell. So this was never discovered before. So I was like very excited to be part of this. And yeah, that was basically like my first paper, this discovery of this helix being able to know, okay, how much cholesterol is in the cell. So that was pretty cool. The other thing I did show as well is this protein being needed to pull out the squalene monooxygenase protein to be destroyed. So what you can see is that normally, if I just have this protein around, you don't see much of it. Like in the cell, there's not much of it around. But if I block this guy, if I block this VCP just by blocking it with an inhibitor, you can see there's a lot of signal. So this protein, this squalene monooxygenase protein, is suddenly accumulating a lot. So that's telling me that this protein is always destroyed, and I can block it from being destroyed. So that was pretty cool. That was published in Biochemical Journal just last year, I think September. Hasn't even been a year yet. So that's pretty exciting. And there's another paper about this as well. So, but I'm not going to discuss that too much. If you want to read more of my stuff for the people who are like more of the scientists, you can look up my Google Scholar account and these sort of stuff on the left here. There's some generic short articles on the right, which we talked about squalene monooxygenase in cancer, if you're interested in that. And yeah, basically each of that schematic I showed you, each of them was published in each part of that schematic was published in a different paper. The first one is about the helix. The second part is about ubiquitin. The third part is about how we pull it out of the membrane. So it's kind of intriguing. We actually didn't think we'll finish that schematic. We thought we would just do one thing and then just ignore the rest, but it just ended up that way. Anyway, cool. So why do we care, I guess, is one of the big questions. You know, you always have to sell your science. And one of the problems with statins is that you can block this, right? You can block this. You can use a drug to block HMG-CoA reductase. But if you block this really early, you can have side effects because there are many important molecules in between that you don't make. So that's very important. These molecules are very important. Now, the other way is that naturally our cells will use lanosterol in the middle here to destroy HMG-CoA reductase. So there's two ways to control cholesterol levels. One is using statins, one is using lanosterol, which is a natural way to stop the activity. So someone has actually used this natural way to destroy it and lower cholesterol levels, and I'll show you now. Oh, actually, I want to tell you one fun fact as well. Like I think Wen was mentioning in her first talk as well, that a lot of our drugs are actually discovered randomly sometimes. You can get first isolation of statins was from Kyoto in a random sample of like mold. And now it's become $1 billion making industry. So there's a lot of nice history behind that. And I really truly enjoy working in this field for that reason. But yes, time to grow some random mold sometimes. Cool. So the problem with statins is that you can see statins, this is this green triangle, right? So statins can bind to HMG-CoA reductase, okay? So it binds here and it binds there, you can see that. But what happens is your body will try to compensate. It will make more HMG-CoA reductase. So when it makes more HMG-CoA reductase, you can see there's more of it. You need to give your cells in your body more statins. So it's like your body is fighting against it. So they become less effective. So one way to control this is by destroying it. So this group in China, which I'll mention later on again, they use this compound 81, and compound 81 actually looks like the nostril. It looks like the nostril. And so basically what they've done is they designed this compound to destroy HMG-CoA reductase by making it more ubiquitinated, joining with ubiquitin, and then it gets destroyed and you lower cholesterol levels. So this is one way for us to lower cholesterol levels, and that's to destroy something rather than to block it. So that's going to be the second part of my talk later on. But what you can see here is that if you have compound 81 or statins, this is a statin, LOVA, you can see there's less red dots. These are actually atherosclerosis lesions in mice. So less red means the lesions is less atherosclerosis. If you combine compound 81 with a statin, lovastatin, you can see there's even less red. So you can make this even more effective. And this is something I think it's really been in hot interest in the past few years. Similarly, and I didn't do this, but I wish I could do this. We know that cholesterol destroys squalene monooxygenase by targeting this region. There's also inhibitors out there that block squalene monooxygenase. So possibly we can do similar things to destroy squalene monooxygenase using something that looks like cholesterol. Let's not put cholesterol into our cell. We can use inhibitors out there that we already have. Green tea polyphenols, which are basically found in green tea, garlic extracts, resveratrol is found in red wine. They all can block squalene monooxygenase activity. I'm not saying you should go and drink a lot of red wine after this, but these have been shown to help lower cholesterol levels. And one possible way is that it's blocking squalene monooxygenase. And again, please don't go take a lot of red wine. You need a lot of red wine. Cool. So there's natural way, and there's also like chemical ways as well. These are chemicals, MB508 and compound 4. Okay, so the reason I want to really make this clear is because cholesterol is actually very bad for many things like heart disease and cancer. High cholesterol levels can drive some cancers. You know, even cancer cells, they need to know how much cholesterol is around. Cholesterol will help to grow cancer cells and tumors. So there's a lot of prospects and opportunities to target this. What you can see here is that in mice, if you have a lot of squalene monooxygenase, the livers of these mice, they have this balloon formation. So these balloons are a sign of liver cancer being worse. So these mice have a lot of cholesterol, and this is due to more squalene monooxygenase in these mice. So these are from liver tumors. I will also mention that this same study, they showed that you can use terbinafine, which is used to treat athlete's foot. They gave the mice terbinafine, and they see less, they see a reduction of the tumor size and volume. This is interesting because terbinafine actually blocks fungi squalene monooxygenase, not human. So that's weird. You know, you've got something that you can commonly buy in a pharmacy or the chemist, I think some people call it. You can get this drug that you use to treat athlete's foot. And the way it works is that it blocks fungi squalene monooxygenase, but then why does it work in humans then? Or why does it work in mammals? So that means it's toxic still. So that's quite interesting in my perspective. So I want to make it clear that we animals and humans, we make cholesterol, plants make something called phytosterols, like phytosterols. Fungi make ergosterol. So terbinafine should only be blocking the fungi, but for some reason, terbinafine is able to block this step here in humans as well, or animals, I should say. But I think you need really high doses, so that's just a precaution as well. So this terbinafine actually binds to this region, the catalytic domain, and this catalytic domain is where it's for the activity. It's found in all kingdoms of life. It's found in plants, it's found in animals, it's found in fungi. So that's pretty exciting and interesting, but we can now use a different drug possibly to target this protein. Terbinafine resistance is actually not so big of a problem, unlike antibiotic resistance or antifungal resistance in Canada. And again, you can hear more about that in another previous talk if you want to, from a woman that was from previously, but she talks more about antibiotic resistance. But basically, you can have conditions where you use too much of this and the fungi on your feet could be resistant, I'm just saying. Some people as well, it depends on the mutations and all these kind of things, but just fun fact. Cool, that was pretty much, I guess, a really broad overview of what I was interested in my PhD. I did a lot of things. I was interested in the protein, I was interested in cancer, I was interested in many, many things. I did a little bit of everything. And when I wrote my final paper in my PhD, it was really a generic, general article that covers all of what I just said. So now I'll be talking about my postdoc, which I started March this year, and this will be very short. It's not going to be too complicated, I hope. But you have the ubiquitin system, right, that I told you just now, where ubiquitin can join to HMG-CoA reductase or join to squalene monooxygenase, and this destroys them. And when this destroys them, you can control how much cholesterol is there. So this is one way that the ubiquitin system can control health and disease. But it's actually more than that. So I'll give you an example. If you have a protein in your cell, it hasn't finished its job. It will stay there. You know, this protein A could be doing anything, but it's just staying there if it hasn't finished its job. Now, these guys are also proteins. They're called E3 ubiquitin ligase. There's 600 different kinds in the human genome, in humans. But say protein A has finished its job. What can happen is that the cell can make the E3 ubiquitin ligase join to protein A, and then this will cause the joining of ubiquitin to your target here. And when this joins, this guy, the proteasome of a protein, comes to destroy protein A. So here it's saying that ubiquitin is like a signal. It's like a name tag that tells you to get destroyed. So it gets destroyed when protein A is not needed anymore. So it's just finished its job. So that's pretty cool and pretty exciting, that your cells can just get rid of something. Now, there's another possibility, right? You have protein B. Sometimes your cells make it and it's not made properly. It can cause a disease or something. And your cells can also bring an E3 ubiquitin ligase to then join ubiquitin onto the target. And then this causes it to be destroyed again. So your cells are very good at getting rid of things it doesn't need. Your body is very good at that. But there are some times where it cannot do that. So if you have a protein that causes cancer, all right, and these two are not matching, you know, and protein C is always around promoting cancer. What we can do nowadays is to design a chemical molecule, which we call a PROTAC. So a PROTAC has a head group that binds to protein C, and then it has a head group that binds to the E3 ubiquitin ligase. You have to design these. And then you design a linker to join these two molecules. So what happens is that you would then force this to form a complex. It will force everything to come together. And this will force ubiquitin to join onto your target. And then when it's joined, it gets destroyed. So here you can artificially destroy a protein by force. So you're hijacking and manipulating your cells to destroy a protein. And what is good is that you can recycle this. The E3 ubiquitin ligase gets recycled, and the PROTAC molecule also is recycled. What does it look like? This PROTAC, it looks like this. It's just all chemical molecules. You have a head group that can bind to a target. You can have a linker, and then you have adapters to bind to E3 ubiquitin ligases. So these are these guys right here. So they're all chemical molecules that we make. So why is it so beneficial to do this? You might think, what is so special about it? traditionally, we use inhibitors to target proteins. So what happens is that when you target proteins with inhibitors, you form, if you have 3 molecules of protein C, you have 3 molecules of inhibitor, that's it, that's finished. You might need to give your cells and the patient more inhibitors, that could be bad. But with a PROTAC, it's different. Once this joins ubiquitin and your target is destroyed, this thing is recycled. So you don't have to use very high doses of this chemical molecule, and it can do the same thing over and over again. And then it gets joined to ubiquitin, and then it gets destroyed. So that's very effective. And it's also good because it's reversible, unlike CRISPR, which is a technique to edit the gene that's permanent. So here we have a potential to mix and match your molecule depending on what you want to get rid of something. So it's like Lego. So I'll just show you a very short clip. I hope it plays. I hope it plays.
Ubiquitin conjugate proteins to the
I don't know where it starts.
disease-causing protein.
So I'm going to show you what the PROTAC looks like in a cell, and this lady will explain it to you. Hopefully it's similar to what I just said.
Machinery and degrades specific disease-causing proteins, including proteins not treatable by traditional drugs. A PROTAC is a small molecule with 2 binding regions. At one end is a region that binds a disease-causing protein targeted for degradation. At the other end is a region that binds to an E3 ubiquitin ligase, part of the natural ubiquitin ligase complex. The regions are joined by a linker that is critical for each PROTAC's specificity and function. Inside a cell, the PROTAC forms a trimer with the target protein and an E3 ligase, directing the ligase complex to tag the targeted protein with a ubiquitin molecule. Acting repeatedly, the target protein is tagged with additional ubiquitin molecules. Once a chain of ubiquitin molecules is attached, the proteasome recognizes the target protein and degrades it. PROTACs act iteratively to tag and degrade additional molecules, leading to the potent destruction of the disease-causing protein. Arvinas.
I'm not promoting any companies or any drugs, I'm just saying. But Arvinas is one of the first ones to actually trial this. The technology actually has been around for a while. It's been since 2001. But it was slow. At first, I thought they just discovered this in the past 5 years because I only heard about it 5 years ago. As you can see, 5 years ago, it just started to ramp up more. There's more people reporting about PROTACs more and more. And I was like, why are we only talking about it now? So it's been there for 20 years, but it's only in recent years that people are trying to develop this. So there's like PROTAC molecules in the clinic now for prostate cancer. And yeah, that's what I'm interested in. Well, the lab is interested in. So what am I going to be working on? I still don't know in my postdoc. I'll be working on a protein X that I don't know yet. I'll be trying to destroy a protein in a disease. I don't know what disease yet, but we know we're focusing on inflammatory diseases, so things that are like inflammation. And we will force an E3 ubiquitin ligase to come and join ubiquitin to destroy the target. So that's the plan. And we'll design a PROTAC for that. So it's a really big ambition that the lab I'm in will have. And that's probably gonna take another, 4 or 5 years of my life, but we'll see. Cool. There's other molecules out there to target the ubiquitin system. I won't mention it, how they work, but there's drugs out there already for multiple myeloma. There's 2 kinds, there's Velcade and lenalidomide, I think, yes. And so there's different kinds of drugs. These 2 have failed clinical trials, I think, but basically I'm just saying that the ubiquitin system is something we can target for disease. There's a lot of E3 ligases, as you can see here, and they're usually, some of them are associated with diseases like Parkinson's, cancer, Huntington's. So there's a lot of disease relevance is the point I want to mention to you. I'll give you an example. You might have heard of Parkinson's. Parkinson's is a neurodegenerative disease that happens as you age. So what is important is this guy called Parkin. Parkin is an E3 ubiquitin ligase. Now, there's something called early onset Parkinson's. So it's people who get Parkinson's disease before the age of 45, I think. So what happens is that these people have mutations in Parkin. So when you have mutations in Parkin, when you have a damaged mitochondria in your cell, the cell cannot get rid of the mitochondria, and because parkin is not functional. So if parkin is not functional, this damaged mitochondria in your cells will accumulate, and that's basically going to result in a lot of symptoms of Parkinson's. So that's not good. So there are people trying to develop ways to activate parkin. And I want to give you an example of an H3 ubiquitin ligase. What happens if you lose it in mice? So these mice have been fed a high-fat diet to make them obese. What you can see is if we lose an H3 ubiquitin ligase, these mice are slightly leaner, smaller in size. Their livers are smaller in size. Their fat tissues are not big and heavy, unlike the control. So basically, the H3 ubiquitin ligases regulates a lot of processes, and that's just kind of what I want to really point out. Yeah, I guess I'll want to give you a little bit of historical context. I think we're almost done, actually. I guess what I've learned over the years is that it's kind of cool working in basic science because you work on stuff that was passed on from generation to generation. So when I talked to you about the cholesterol synthesis pathway, that was discovered by Nobel Prize laureates, Nobel laureates Conrad Bloch and Phil Linen. This Japanese scientist, Teru Ono, worked with them. And he went back to Japan and later on worked with other scientists in Japan. And I didn't realize it, but it was last year when I started to write reviews. In the past few years, I've written reviews, but squalene was first obtained from sharks in Japan. So there was a lot of history of Japan and squalene and cholesterol. And I was like, oh, okay, that's cool, that's cool. Then I went to Japan last year for the first time, and second time, but for work for the first time. And I met these people who I didn't know they were associated down this long, long family tree. But it was really exciting talking to them, learning about history of like squalene, and like basically it's like seeing my research in paper coming to life when I go there. It's like, your experience, and Japan has a lot of nice fish. So it was really exciting to be able to explore this history, like personally. And squalene monooxygenase, like I said, squalene monooxygenase is found in all kingdoms of life. In plants, we make phytosterols. You might know phytosterols as something that you see in your margarine or even supplements. Some people sell it. So there's a lot of history behind squalene and phytosterols as well. But what I want to point out is that squalene is actually useful. Squalene is the thing that squalene monooxygenase uses. So squalene is found in supplements. People sell it. I'm not saying, I don't know if it actually does anything, but I know in terms of a vaccine, it's actually very useful to boost the efficacy of the vaccine. So Fluad is actually a flu vaccine developed by Novartis, and they have squalene in there. Cosmetics, people actually use squalene to make a different lipid called squalene, and they use it in cosmetics, which is pretty cool. And fun fact, if you're born in Southeast Asia, chances are you would have come across ginseng. And the cholesterol synthesis pathway in ginseng is actually, that's why I mentioned plants earlier, because squalene is actually a very central piece of life that can go down to make these things called ginsenosides. And ginsenosides are actually the active compounds of ginseng that makes them that's responsible for the health properties of ginseng. So there are researchers who work on metabolic engineering, and that is to engineer these pathways to make them more active. In yeast, for example, so you don't have to get the ginseng from ginseng. So you don't have to get the ginsenosides from ginseng, you can get it from yeast. So it's like biotechnology applications here. So this is playing around with the proteins, which is kind of cool. In terms of the Nobel Prize stuff for cholesterol, there's also Brown and Goldstein. My supervisor, Andrew Brown, has worked with them in 2000, I don't know, early 2000s. And they trained a lot of other scientists. And so I worked with Andrew afterwards. And it was really interesting hearing about all the Nobel Prize stories from him and how the history of cholesterol has developed. And I think that's what I really enjoyed during my PhD. It's getting a historical context, not just like, oh, I'm learning about what's in the past 3 years, but you learn about history, you learn about how science is developed. So it's really interesting. And I really appreciate that. Ubiquitin also has a Nobel Prize. So unfortunately, I'm not too familiar with that side of things. I'm going to stop there because I feel like I can say any Nobel Prize is associated with my PhD. I can say green fluorescent protein, I can say anything, but those three were the main ones that are related to my PhD, and I really enjoy working with these areas. And I guess, I just want to mention really briefly now where I am, where I came from. So I was born and bred in Malaysia. I was raised in Miri. I did my secondary school in 2 schools, SMK St. Joseph and SMK Baru. This is where I did my Form 6, actually, halfway. So halfway through Form 6, I realized that it was too hard. It was not quite for me. It's a high school exam we have there. Even if you ask me to go back and do it now, I don't think I'll get an A. Even I have a PhD and I don't think I'll get an A. I think Sarah did Form 6, I'm pretty sure, and she did it all the way, but I'm not that smart. And I realized I wanted to do life sciences, or I want to be a vet. But I realized in Malaysia, the options might be too limited. And I was like, what can I do? What can I do? What can I do? And it was too hard, I was struggling really hard for half a year. So then I decided to go to Riam, which is also in Miri, to do A-levels. It was private, the Cambridge A-levels. It's private, it's slightly more expensive, but I was like, well, I'm going to take a risk. I'm going to study hard, get good grades so I can get a scholarship or something like that. So that's what I did. Unfortunately, I didn't get a scholarship, even though I got straight A's, so whatever. So then I went to Inti, which is in Nilai, very isolated place, and can get kind of peaceful every now and then. Lots of mosquitoes, I think. And so over there, I did a program called Australian Degree Transfer Program, where you can transfer to partner Australian universities. So the plan was, if I don't get a scholarship from these universities or any of them, I'll go to the cheapest one, and that was the University of Adelaide or University of Queensland. Or if I really am kind of like not really liking the whole financial situation, 2013 was a very expensive year because the Australian currency was one of the highest exchange at the time. Well, that's a long time ago. Anyway, so if I didn't get a scholarship anywhere, I was going to stay in Inti, do the biotech program there, and just finish in Malaysia. It's not a bad place, But then I got a scholarship. I got a scholarship from UNSW, which was $70,000 for tuition fees for 2 years, and that's a lot of money. But then there was about 2.5 years I needed to study in UNSW. So I paid for half a year's remaining by myself. So I lived in Sydney for 7 years. And in Sydney, the bachelor program is 3 years, or you can do 3 years and 1 year, so honors research years. So I did this with Andrew, my PhD supervisor. But this was a very sensitive period because what other speakers get often is what do they want to do in the future, right? So I knew what I wanted to do in my future already. I knew I wanted to live in Australia ever since I had that opportunity to go to Australia. I was like, oh, it's so nice to live there and stuff like that. And money is great. But then I realized it was a long way to get there. And I knew that I needed to get a good honors year to get any chance at all to do a PhD, to get a good scholarship for a PhD. So I need good grades for my honors research year to get to a good PhD project. And a good PhD project with good outputs and good papers will likely lead you to a good opportunity for a postdoc job. And then that leads to an opportunity to, publish and whatever. So it was like this long train of thought. I don't regret my decisions at all as to who I chose for my PhD. I think I chose the right one, and I'm in a good place for doing my postdoc as well. Yeah, a lot of it was a bit of luck. And now here I am in Melbourne, which is the naughtiest place in Australia because we have the most COVID cases right now. I think I moved 1 week before the city went on lockdown, so it was fun. Cool. I guess a lot of my decisions in my PhD, even I guess especially doing honors, was like I knew where I want in life. I knew where I wanted to go in life, so it was very goal directed. So a lot of the times I don't get what I want along the way, but I choose the next best thing, and sometimes the next best thing was actually better. For example, I could have done a postdoc in Sydney, where a lot of disastrous situations are happening for many institutes, but I didn't. I moved to Melbourne instead. The other dilemma I always had was, should I do basic science or applied science? And I think Shuya earlier, or even when you guys were discussing in previous sessions, that we like basic science, we like to enjoy what we do, but sometimes it's really hard to survive in it. And there's been a lot of times where I go based on opportunities. Sometimes I go based on interest. It's great if you can have both, it's great if you can do both. But I realized it was always this toss between, do I like what I do? Do I do what I like? There's been a lot of that decision through my whole PhD, and even until now. But I think you choose based on the situation, and again, the next best thing might also be better than the first option. How is the day in the life of a scientist? Sometimes a long day, some 14-hour days, you have random incubations that just, you gotta stay there. Some days are short, like 6 hours. Lots of manual labor, you can actually lose weight from it. I know my friends who are not scientists think that, oh, you just sit around the computer, you don't do much. But no, you can actually lose weight, trust me. If you do a lot of work, you can get papers and data, you can go to conferences. That's just my stuff in the lab. I do a lot of Western blots. I do a lot of cloning, which is a lot. That's just needles for preparing proteins. You go to conferences, you give talks, if you get the chance. These are rare if you don't have data, and depends on your supervisor. You meet Nobel Prize winners, which is kind of cool. This was me with Lee Hartwell in San Diego. He was pioneering the work in the cell cycle, so that was pretty cool. I didn't even know I was going to meet him. Cool. What I like about my job, I really like going to all over the places in the world. And this is actually quite hard because you need to get your own money, usually, or your lab pays for it. And I was lucky because I got a lot of sources of money from outside. So I got to go to China for the first time ever as a tourist and also to work there. So that was pretty cool. I've never been to China until last year, way before the pandemic, just saying. And I got to go to Korea, which is also very cool. I've never been to South Korea. Sorry, I have, but with my family. But on your own, traveling for work. I got to go to Japan. Again, all this money is sourced externally. It was like through scholarships or travel awards and things like that. And it's really exciting because if you like anime, and then I went to this staircase, very famous one that was shown in this anime movie, Your Name, and I was like, I gotta find this staircase. So it's really cool being able to work and travel, but more importantly, being able to work and travel, I get to experience what it's like living over there, not just for as a tourist, but for work as well. And you get to engage with people all over the world, different scientists, for longer periods, like 3 weeks or something like that. So I really like that about my job. But again, it's really subjected to if you have money to go, you have data to present, or you have collaboration. So that's what I really enjoyed. And again, I really thank my PhD supervisor for giving me the chance to even go. He has to say, yes, you can go, then you can go. Yeah, I think acknowledgements, endless, really. I think it's just, family for really just believing me blindly, not knowing what I do. They just believe you, like you're doing what you're capable of taking care of yourself, so whatever. Friends, I think, are very important because friends are who shapes you in like your PhD personalities. Your colleagues help you with that, but I think the soft skills you learn mostly from your friends and the social support. And they don't train you as a scientist. Your friends can't train you as a scientist. Your colleagues and your mentors do that. But I think collectively, that's what really made my PhD, I think, fruitful and worth it. And yeah, I think that's pretty much how it went. Yeah, I guess I just want to thank you for listening. I have a blog. If you want to check out my blog, I have a Twitter account for my science. Otherwise, you can always just reach me somehow, somewhere on the internet. I'm pretty sure you can find a random contact. But yeah, that's pretty much it.
Yeah. I learned so much from your presentation. And yeah, we've got a few questions, actually a lot of questions. I'm going to ask about cholesterol first, and then ask a bit about ubiquitin, and then about the PROTAC side of things. So so basically your presentation was just showing us that cholesterol regulates itself, basically.
Yes,
cholesterol.
Yes.
Oh, wow. Okay, so that answers Tom's questions. And you said like 70% cholesterol is made in the liver, which is quite surprising because I always thought that we always consume cholesterol and that's the only way to get it. So my question is, are there many cholesterol diseases that are due to genetic mutations in squalene monooxygenase? Do you know?
So cholesterol-related diseases in squalene monooxygenase.
Yeah.
No, unfortunately. So a lot of people study squalene monooxygenase in the context of cancer and actually Rett syndrome, which is an autism disorder, autism spectrum-like disorder. No one's really looked at SNPs or mutations in squalene monooxygenase. It doesn't seem like it's correlated with heart disease that strongly. There's been some suggestions, but no one has really looked into that. Some things are stronger biomarkers than others, that's for sure, but not squalene monooxygenase.
Yeah, so are most people with, obesity is like a big issue in Asia. I was just wondering if whether most people with high cholesterol levels are due to like genetic reasons or basically their diet.
Yeah, so I think the dogma has changed. You know, your aunt and uncle might say, "Oh, don't eat so much eggs" or whatever. But it's more than that. There's actually a lot of diabetes connection as well. So carbohydrates it contributes a lot to obesity as well. So that can result in higher cholesterol levels. Other lipids, other fats can also do that. It's not just cholesterol itself. So cholesterol is sometimes secondary. It's not like the primary thing as well. And you might have heard of a lot of random diets over the years, the ketogenic diet, 5-2 diet, blah, 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 all these kind of different fat diets. And a lot of them actually work by affecting calorie intake and what nutrients you take. And cholesterol has never really been a key component in any of these things. So it's always been the carbs or other lipids, other triglycerides. So cholesterol is almost like a later on thing. So you have to control other stuff first. Yeah.
So you know the cholesterol production that you talked about, the pathway. Yeah. So we know there's bad cholesterol, which is low density lipoprotein, and then good cholesterol, which is high density lipoprotein. So is that a post modification of cholesterol or?
So the thing is, well, is that we cannot. How would I say? I'm just scrolling as well for other stuff. So the cholesterol that you get from your diet or you make it yourself, it goes into lipoproteins, right? So lipoproteins are like circular molecules that contain cholesterol and protein. They are just simply stored forms of cholesterol. Your body will eat it and then it gets transported in your blood in these lipoproteins. So LDL is from your liver to the rest of your body, usually. HDL is from your heart back to your liver. So the reason HDL is good cholesterol is because you need it to transport excess cholesterol from your heart away from your heart.
Oh, wow.
That's why you need HDL, whereas LDL is for kind of like the rest of your circulatory system. So it's just a stored form of cholesterol.
Oh, nice. I didn't know.
Yeah.
Oh, cool. Very interesting. So we have a question from Wun. So she was asking if HMG-CoA reductase, whether it's involved in other signaling pathways or is it just involved in metabolic pathways?
It is pretty much the enzyme itself is very important in metabolism, but the pathway is regulating a lot of processes: immunity, cancer, Alzheimer's disease. So there are some people who, this is actually on the scientists as well, there are some people who think statins can be used to treat cancers or even treat some autoimmunity disorders or autoinflammatory disorders. So by affecting HMG-CoA reductase, you can affect a lot of other pathways as well. I shouldn't say this, but there's some people, again, there are some people who are like, oh, maybe statins can treat some virus infections because low levels of cholesterol from statins can actually boost antiviral immunity. Yes.
It's a selling point for media, but it's the wrong selling point.
Yeah. So people are repurposing. The statins have been around for so long that people realize, hey, hang on, the side effects from statins is actually beneficial for some other stuff as well.
Interesting. Yeah. Cool. So we'll move on to questions about ubiquitin. So Julian from Facebook. So she's asking how many ubiquitin molecules are required to trigger the process, as in polyubiquitination. So is there an effect when it's monoubiquitination?
So I actually don't know. Each PROTAC is slightly different and each ligase is different. But normally in a cell, you need like 4 ubiquitin molecules to be joined. Like, so 1, 2, 3, 4, and then it gets degraded. So that's usually how it is. And she also said Melbourne has the best coffee, and I agree.
Yeah. So we have another question from Hakim as well. So would the cells prefer is there a preference for monoubiquitination or polyubiquitination, or is it?
I think it depends on the PROTAC, the cell, and the E3 ligase, to be honest. Yeah. At least with the E3 ligases I work with, it's usually polyubiquitins, which means a lot of ubiquitin joined onto it. Monoubiquitin, which is 1 ubiquitin, I don't think it's such a strong signal for destruction. It's dependent on some cases as well, some proteins only. Yeah.
Right. So you're saying that polyubiquitination is a better tag for protein degradation.
Yeah. At least that's what people think.
Right. So a question from woman again. So is the helix that you mentioned required for squalene monooxygenase? Does it require it to function normally?
In a way, yes. So that was the purpose of my PhD, to understand how regulation affects activity, because you don't want activity to go uncontrolled. So yes, it does control the activity, even by a bit, to I don't know how many percent, but it does. Yeah.
Yeah. So a question from Jia about statin. So can statin cause an accumulation of acetyl-CoA, and is there a side effect to that?
So I don't know. I don't think it can, and I don't think it should, because acetyl-CoA is a substrate for other biochemical pathways. As soon as you accumulate acetyl-CoA, it will get shunted into other biochemical pathways. And I don't remember what acetyl-CoA is used for as well, but other metabolic pathways. So it shouldn't cause the accumulation. It will just get metabolized.
Oh, cool. So Julian again from Facebook. So you talked about green tea extract, compound 4, and garlic as to block squalene monooxygenase, right? So what's the common feature among them? So is it the structure of the chemical itself that blocks squalene monooxygenase?
So yeah, I'm seeing that as well. That's a good question. That's going back to literature in the 2000s, which sometimes I forget. So some of the compounds like garlic, it's because it can react with the cysteine residues of the catalytic domain. Compound 4 and then the chemical compounds, so the natural inhibitors, what people think is that it affects the cysteine residues. It binds to and reacts with them. The chemical compounds, on the other hand, they actually bind to where the substrate binds exactly. So one is that they bind to substrates, and one is that it's simply just messing around with the cysteine residues in the catalytic domain somehow. Yeah.
So it's not the structure itself, but messing with the.
Yeah, it somehow can bind there, and they think that it binds there and affects it somehow.
Right. So again, another question from Julian from Facebook. So she asks, is terbinafine? Terbinafine was shown to be effective in humans, right? Is the lipid composition of fungi, squalene monooxygenase, similar to human SM?
So they use that terbinafine in mice, which shouldn't actually have any effect. Because first of all, terbinafine blocks fungi squalene monooxygenase, not mammalian one. So the lipid composition of humans and fungi are very much different, whether it's in the bilayer or wherever. And the most notable difference is that humans make cholesterol, fungi make ergosterol. So that's the main difference already. So the reason that it's effective in human is because the very, very, very high dose that they use can just force terbinafine to enter squalene monooxygenase binding pocket. It's not ideal. So the binding is not ideal, but when you use really high doses, it forces it in there. So yeah, although you can use it to treat your athlete's foot, you'll never use it in high enough to be toxic to you.
It won't pass the FDA health and safety.
No, it wouldn't. It's already been FDA approved. Yeah.
Yeah. So I will move on to questions about PROTAC, which is quite cool because I've never heard of it, but it sounds really amazing. Tom asks, how do you actually introduce PROTAC into cells?
You just add them into your cell.
Oh, do you inject them, or do you just put it into solution and the
you just
cells?
Put it into solution and then the cells will take it up.
Right.
Some of them are harder to go into cells than others. The linker can affect it, the size of the head group can affect it, but generally the new ones, the ideal one is it's got good pharmacokinetics. You can just add it, it goes into the cell. That's it.
Yeah, but that's for a lab environment. How about a clinical environment? If PROTAC were to.
Yeah, so the idea is you can give it orally administer, or you can just inject it. So it should just be soluble and stuff like that, ideally. Yeah.
Right. So it's okay in the gut acid then, I assume.
It should be. Yeah, it depends on how you deliver it, obviously. We haven't gotten that far yet in Australia, but that's what we're working on, I guess.
Yeah. And yes, so I have a question about PROTAC. So will PROTAC be considered a personalized medicine then?
It definitely won't be personalized, because I guess personalized is always like about the genome and genes and things like that. This is more about being able to expand your tools to treat diseases. Previously, with normal inhibitors, there's a lot of pathways we cannot block because we need high doses or whatever. And PROTAC is really cool because you can mix and match freely to destroy anything you want. You just need to find a chemical that binds to it. So it's not personalized, but it's a way to treat more difficult diseases that previously have not got good treatments. Yeah, that's the aim of it. You can even degrade transcription factors as well. So it's just anything really.
Yeah. So you talked about the advantages of PROTAC. Are there any disadvantages as well?
The disadvantages.
Or are you biased?
No. There's actually disadvantages. You can Google it, and there are. So one of the big disadvantages is the combinations of head groups that you need to try and mix with chemical biology. So chemists need to keep synthesizing different kinds of linkers, screen here, screen there, until you find something that binds comfortably. And you also rely on the endogenous, so the levels of the E3 ubiquitin ligase in your cells. So if that cancer doesn't have the E3 ligase, then you have to swap. So it depends on manipulating the normal system to degrade something. So it's not, you're relying on something else. Yeah, there's something out of your control as well.
Cool. So Julian again from Facebook, which component of PROTAC is most important to drive ubiquitination and thus, degrading the protein? Is it the linker or the head of the protein binding domain?
I think it's actually everything. People sometimes can get a PROTAC that doesn't work because the linker's not working. You can have a PROTAC that doesn't work because the head group that binds to the E3 ligase is not working because the cells don't have the E3 ligase. So it's really about mixing and matching that three parts until you find the ideal one. So it's really all together. That's why we're comparing a lot of them.
Right. So we've got quite a general question from Hakim. So about media, have you yourself personally been frustrated with the media taking science out of context?
Yeah, I have several times, and that's why I wrote a blog. I started a blog for that reason as well, to give my own personalized insights into science and life.
Yeah. Cool. So read Jake's blog if you're interested about his frustrations. That's I think the blog is where Jake vents all his frustrations and rants about life. So a quick question by Tom. Do you recommend a PhD that's more broad or too focused on one specific area?
It's always one of those things. Do you become a master of none, or do you a jack of all trades master? It's always a tough one. And I finished my PhD, and I'm on team. Be a jack of all trades and master of none.
Okay.
When you're talking about techniques, when you talk about techniques and experiments, be a jack of all trades and master of none. You can learn a little bit of everything. That's fine. But in terms of knowledge into a specific field, then you really have to master your knowledge in that field. There's no point learning about cancer this week, dementia next week, and the next week you learn about like genome editing. You just don't have that space. You have to really be an expert in your field. But in terms of the techniques, it's like just learn as much as you can. But knowledge-wise, you really have to know your stuff.
Otherwise, I guess that's also dependent on what you want to do after. If you want to do like an academic, if you want to pursue a career in academia, then you have to be an expert.
Yes. And I guess also skill sets wise, not just experiments, but the soft skills, it's definitely like better to just learn a whole bunch of things. Then you find out what you're good at.
Yeah, so we're low on time, but just one last question to wrap everything up. So what's next after your postdoc? Any thoughts about that?
Just started my postdoc. I don't know. I don't know. I don't think I'm quite there yet. I think I'll see how I go, because when I was doing my PhD, what prompted me to do a postdoc as well was because I got papers, I was on a good track record, I was going to conferences, and there was a lot of encouragement for doing a postdoc. Everything was going according to plan. Everything was going in the direction that's like, oh, go do a postdoc. So I guess it depends on what happens next. If for the next 4 years I get nothing out of it, it's just not worth it or whatever, then, I have to find something else.
Right, so your contract is 4 years then?
I don't know. I don't know.
Anyway, thanks for your time, and I guess good luck to your new postdoc job.
Yeah.
Right, I think that's all the questions that we have. I think, Jake, if you have time to share about the work culture in Australia, do you have a blog piece about that?
No, I think it's really hard. You can, whatever I can say on my blog, you can easily Google as well. Just like Google, how is it working in Australia, blah, blah, blah. It's usually quite accurate.
Right then. So thanks, Jake, for your time, and thank you everyone for joining us on a Saturday night or afternoon for those in the UK. Yeah. So we have our next speaker for next Saturday, who is Shiman Yi. She's a research assistant in Cancer Research Malaysia, and her research team aims to identify new therapeutic targets for head and neck cancer. So for more updates regarding future Research Insider series, like our Facebook page and Bios and Harder Scientists of Malaysia. And yeah, I think that's it. Thank you, everyone, and see you next Saturday. Bye.